Hello, welcome back to our video tutorials for biochemistry. Today we're going to look at a search tool called BLAST, which is a sequence search tool to find similar proteins. So the first thing we're going to do is go to BLAST, and I'm just going to type in NCBI BLAST, and the first hit, BLAST Basic Local Alignment Search Tool. Once you get to the BLAST homepage, you need to first select the type of BLAST search that you want to do. So we have options for nucleotide, protein, translated nucleotide. We want the basic protein BLAST search. So I'm going to click there, and now we're presented with the form to start our search. And the first thing that we need to do is supply the FASTA sequence for the protein that we're interested in. So in order to get that, the most efficient way to do it, I think, is to go to the protein data bank, search for the protein that you're interested in. In my case, it's 4R78. I'm going to click on the link for display files and then FASTA sequence. So here I have the header, which tells me that I'm looking for the sequence of 4R78 chain A. And then I have the amino acid sequence of my protein. One thing that I would like to point out, and this is important for BLAST, but not so much for the other databases that we'll be using. At the very beginning of the protein sequence, or at the very end, there will typically be, let's say, 5 to 20 residues that are used for purification of the protein. In this case, we have a his tag. So in the middle of this stretch, we have 6 histidine amino acids. Those amino acids are not native parts of the protein. They are simply to make purification of this protein easier. So these residues are not important for the BLAST search. They're not part of the native protein. We don't want to search with them. So I'm going to select all the residues that aren't the his tag. And I'm going to copy those. I'm going to go back to BLAST and paste them into our sequence search box. Once we have the sequence that we want, the next thing that we need to do is select the database that we want to search. So here we have options for all the different databases that are available. Typically, the non-redundant protein sequences, or NR database, is the most reliable. It's the best. If you want to limit your search to only sequences that are in the protein data bank, you can do that by selecting the PDB database. I'm going to leave it with the non-redundant database. And then the last thing that we want to do is select the type of algorithm that we want to use. BLAST-P is the standard, very basic BLAST protein sequence search. That's fine for most things. My personal preference is SciBLAST, the position-specific iterated BLAST. With SciBLAST, you can do one search, and then you can select a group of proteins, and then carry out another iteration of searching with that new group of proteins. And you can continue doing that. There's also Delta BLAST, which I'm not too familiar with, but is a newer algorithm and claims to be faster. I'm just going to go with my standard SciBLAST at this point. So I'm going to click BLAST. This can typically take minutes, so while the screen is updating, one thing that I want to point out is this image that pops up, and I like to open it in a new tab so that I can continue looking at it while the search runs in the background. So while the search is running, let's go ahead and have a look at what this figure tells us. So if I go to the next tab, we can see that we have specific hits. And here we have choline kinase, which is what we know our protein is, and here we have all the information that is available for that choline kinase family just by hovering over the bars. You can also look at active sites. So if you hover over the active site line, you can see which residues are considered to be part of the catalytic site or the active site of the enzyme. You can look at dimer interfaces, ATP binding sites, substrate binding sites. Okay, let's go back and see how our BLAST search is coming along. So it's finished. Now, I picked a very well-known and very well-studied protein family, the choline kinases. So all of my top hits are very high quality, and that's what this graph shows you. The scores are all greater than 200, so everything here is red, and the fact that the bars go from this far left side to the far right side means that the alignment covers the full sequence that I entered. If there were any green or blue or black bars, they would typically be much shorter. They might only take up a small fraction of the space on one side or the other or in the middle. They indicate small sections of low quality hits, and we're not too interested in those. So now if I scroll down, I can see all of my hits. There should be, well, there are quite a few, but all of them are choline kinases. So what I'm interested in is this first column. It says that it's a choline kinase from Streptococcus again. I have the scores, the coverage, and then E scores, or the E values. The E values are the expectation values, or the expect values, and they're one of the most important metrics that you can use to judge the quality of your alignment. So the smaller the E value, the better it is. And typically this number is E to the minus something. So E to the minus 50 or E to the minus 60. The fact that this one in particular is 0.0 means that it is very good and it can't get any better. We can also see by the identity. So the amino acid identity, the number of, or the percent of residues in my query structure and my hit structure that are identical. So in this case, they are 100% identical. This protein is exactly the same as the one that I used in my search. So if I click on the choline kinase, I can come down and I see the alignment with the query and the subject or the input. And in this case, again, 100% identical, so I have no differences between these two proteins. As I mentioned earlier with the PSI-BLAST, or the position-specific iterated BLAST search, I have these checkboxes here on the far right side, and they are all checked at this point. I can turn them all off, or I can turn them all back on. I can turn them on and off individually. So if I go down to the end of the search results, I will see an option to run a second iteration of PSI-BLAST using the 500 best hits that I have. So to summarize, when using BLAST, you need to input your protein sequence. You want to choose BLAST or PSI-BLAST. When you get your results, you want to look at the E scores, the percent identity, and then the overall scores. You want to look through all of your hits and get a feeling for what kind of proteins or what kind of enzymes they are. Thank you for watching.